Running low on glucose, does your brain shut down? Or does your body have a backup plan? For the MCAT, you need to know exactly how blood sugar is maintained when you're fasting, stressed, or just between meals. In this video, we'll connect gluconeogenesis and glycogen metabolism step by step, where they happen, how they're regulated, and why hormones like insulin and glucagon flip the switch. By the end, you won't just memorize pathways, you'll understand how they work together to keep you alive and help you crush those biochem questions. Gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors, primarily in the liver, and is essential during fasting. When blood glucose levels drop, such as during prolonged fasting or intense exercise, the body needs a way to maintain glucose supply for tissues that depend on it, especially the brain and red blood cells. The major substrates for gluconeogenesis are lactate from anaerobic glycolysis, glycerol from triglyceride breakdown, and glucogenic amino acids like alanine. A helpful mnemonic is LAG, lactate, alanine, glycerol. This pathway essentially reverses glycolysis, but must bypass its three irreversible steps using unique enzymes. Pyruvate carboxylase and PEP carboxykinase convert pyruvate to phosphoenolpyruvate. Fructose, 1,6. Bisphosphatase bypasses phosphofructokinase, 1. And glucose, 6-phosphatase, converts glucose, 6-phosphate, to free glucose. The MCAT loves to test these bypass enzymes, so memorizing them is high yield. It is also important to know that gluconeogenesis is energy intensive, requiring ATP and GTP, which typically come from fatty acid oxidation during fasting. This connects to the broader metabolic shift toward catabolism when insulin is low and glucagon is high. Clinically, deficiencies in enzymes like glucose, 6-phosphatase, can lead to hypoglycemia, highlighting how critical this pathway is for maintaining blood glucose homeostasis. Glycogen metabolism includes glycogenesis and glycogenolysis, which store and release glucose in response to hormonal signals. Glycogenesis is the process of storing glucose as glycogen, primarily in the liver and skeletal muscle, when glucose is abundant, such as after a meal. Glycogenolysis is the breakdown of glycogen to release glucose when energy is needed. The key enzyme in glycogenesis is glycogen synthase, while glycogen phosphorylase is the key enzyme in glycogenolysis. A useful way to remember is synthase stores, phosphorylase freeze. In the liver, glycogen breakdown helps maintain blood glucose levels, whereas in muscle, glycogen is used locally for ATP production during contraction. The MCAT often tests the difference between liver and muscle glycogen, especially noting that muscle lacks glucose, 6-phosphatase, so it cannot release free glucose into the bloodstream. Hormonal regulation is crucial. Insulin promotes glycogenesis, while glucagon and epinephrine stimulate glycogenolysis. This ties into signal transduction pathways, such as CAMP and protein kinase A, which phosphorylate enzymes to activate glycogen breakdown. Understanding these pathways helps you integrate metabolism with endocrine regulation, a common MCAT theme. The regulation of gluconeogenesis and glycolysis is reciprocal, ensuring that both pathways do not operate simultaneously. This prevents a futile cycle where glucose is both synthesized and broken down at the same time, wasting energy. The key regulatory step involves the enzymes phosphofructokinase 1, or PFK1, in glycolysis, and fructose 1,6-bisphosphatase in gluconeogenesis. When PFK1 is active, glycolysis proceeds, and gluconeogenesis is inhibited. When fructose 1,6-bisphosphatase is active, gluconeogenesis proceeds. A major regulator here is fructose 2,6-bisphosphate, which activates PFK1 and inhibits fructose 1,6-bisphosphate. Insulin increases levels of fructose 2,6-bisphosphate, promoting glycolysis, while glucagon decreases it, promoting gluconeogenesis. This hormonal control reflects the fed versus fasting states. The MCAT frequently tests this concept by asking which pathway is active under certain hormonal conditions. Additionally, ATP and citrate inhibit glycolysis and favor gluconeogenesis, signaling that the cell has sufficient energy. This connects to broader metabolic integration, where pathways respond dynamically to energy status and hormonal cues. Hormonal regulation by insulin, glucagon, and epinephrine coordinates gluconeogenesis and glycogen metabolism to maintain blood glucose levels. Insulin, released in the fed state, promotes glucose storage by stimulating glycogenesis and inhibiting both glycogenolysis and gluconeogenesis. In contrast, glucagon, released during fasting, has the opposite effect. It stimulates glycogenolysis and gluconeogenesis in the liver to raise blood glucose. Epinephrine also promotes glycogen breakdown, especially in muscle, during stress or exercise. These hormones act through signaling cascades, often involving CAMP and protein kinase A, which lead to phosphorylation or dephosphorylation of key enzymes. A helpful MCAT tip is to remember that phosphorylation activates glycogen phosphorylase and inhibits glycogen synthase, promoting glycogen breakdown. This ties into broader endocrine topics, including second messenger systems and receptor signaling. Understanding these hormonal effects helps you predict metabolic changes in different physiological states, a common type of reasoning question on the exam. Glycogen storage diseases and enzyme deficiencies highlight the physiological importance of gluconeogenesis and glycogen metabolism. These disorders result from defects in enzymes involved in glycogen synthesis or breakdown, leading to abnormal glycogen accumulation or impaired glucose release. For example, von Gierke disease is caused by a deficiency in glucose 6-phosphatase, preventing the liver from releasing free glucose into the bloodstream, which leads to severe fasting hypoglycemia. McArdle disease involves a deficiency in muscle glycogen phosphorylase, resulting in muscle cramps and exercise intolerance because muscle cannot effectively access its glycogen stores. A useful MCAT strategy is to link each disease to the specific enzyme and tissue affected. These conditions demonstrate the importance of maintaining proper glucose homeostasis and how different tissues have specialized roles in metabolism. They also connect to broader topics like energy metabolism, liver function, and genetic inheritance. The MCAT may present clinical scenarios involving hypoglycemia or exercise intolerance and ask you to identify the underlying metabolic defect, so understanding these diseases reinforces both biochemical pathways and their physiological consequences.